Thank you everyone for joining us today. My name is Annie Dutcher. I'm the Programs Manager with Free to Breathe, and I am pleased to welcome you to today's webinar, Recent Research Discoveries, What They Mean for Patients. During the presentation, all participants will be in a listen-only mode. If you do have a question during the presentation, please use the chat feature located in the lower left-hand corner of your screen. We're going to welcome questions throughout the presentation for Dr. Suresh Ramalingam, so please feel free to post your questions in that chat feature, and we'll try to address as many of them as possible when we reach the Q&A portion of today's presentation. I want to give a huge thank you to our supporters of our webinar series, Lilly and Boehringer Ingelheim, who made today's webinar possible. It's an honor to be joined by our expert presenter, Dr. Suresh Ramalingam of Emory University. He also serves as the Vice Chair of our Scientific Executive Committee. Dr. Ramalingam will cover a number of items related to lung cancer research advances, as well as addressing Free to Breathe's new research grant program direction, Preventing and Stopping Metastases. Following the presentation, there will be time for a Q&A with Dr. Ramalingam, and as I stated earlier, please feel free to address your questions as they come up throughout the presentation in that chat feature located in the left-hand corner of your screen. And now I want to turn it over to Dr. Ramalingam.
Thank you, Annie, and good evening, everybody. I would like to thank you for participating in today's event. My goal is to go over some of the recent developments in lung cancer and give you an update on what they really mean for patients and caregivers that take care of patients with lung cancer. The good news is we've seen remarkable advances in our understanding of the biology of lung cancer, and we've had targeted therapies approved for lung cancer in the past few years. So I'm going to go over some of these developments in the next 30 minutes. In terms of disclosures relevant to my presentation, I would like to say that I have served as a consultant for drug development to several pharmaceutical companies that have interest in lung cancer, including the two sponsors for this program. However, I don't feel that we're going to talk specifically about a product that causes a conflict that might be material to this. Now, if we look at lung cancer, just about 10, 15 years ago, we had a lot of major problems in terms of how we approach the disease. The first one was the fact that by the time one diagnosed lung cancer, it was stage 4 in vast majority of the patients. And we always thought that lung cancers were related to smoking cigarettes, so every patient was either a current smoker or former smoker, and we did not recognize the entity of lung cancer in patients who never smoked cigarettes. There was also a debate not too long ago whether chemotherapy even had a role in treating lung cancer patients. Did chemotherapy help patients? Should we be giving patients treatments? So that was all in the realm of conversations among patients, among physicians, and the researchers. But then we moved to the era where we now have very good treatment options available, albeit we still have a long ways to go because we're not curing a majority of our patients with lung cancer, but clearly there have been very positive developments as you can see on the slide. One is we know that platinum combining chemotherapy regimens are associated with improvement in survival for patients with advanced stage disease. And not only do they improve survival, they also improve symptoms and quality of life. There have been a number of studies that have been done that have established these facts. We also know that if a patient cannot tolerate chemotherapy or develops progression, in other words, the cancer responds initially to treatment and then it stops responding, we introduce more chemotherapy or alternative chemotherapy drugs, which we call as salvage therapies or second-line therapies. And these drugs have also been shown to improve survival outcomes for patients with advanced stage non-small cell lung cancer. More recently, we've seen the emergence of newer chemotherapy drugs, and some of these drugs have significant advantages in terms of side effects profile, in terms of how frequently they are given, and so forth. So we have a variety of options to choose from when it comes to chemotherapy for patients with lung cancer. We also know now that there are different subtypes of lung cancer. Small cell lung cancer, which is decreasing accounts for about 15% of all lung cancers, and non-small cell lung cancer, which accounts for the remainder 85% of patients with lung cancer. For today's talk, in the interest of time, I'm going to focus primarily on non-small cell lung cancer. Now, non-small cell lung cancer also can be divided down into adenocarcinoma, which is by far the most common, then squamous cell and large cell. Those are the three main types of non-small cell lung cancer. About 10 years ago, we treated all subtypes of non-small cell lung cancer with very similar chemotherapy regimens or treatment approaches. But now we understand that these individual subtypes have different biological behavior, and so we start treating them each as individual distinct entities. So that's also a significant step forward in treating lung cancer. Now, if we just look at past 5 to 10 years, what we have learned in lung cancer, these are highlighted in the slide here. We now know that patients who've never smoked cigarettes and get a lung cancer, that's about 10 to 15% of our patients, have very distinct biology. They are not the same as somebody who's developed lung cancer after tobacco exposure. And we have to recognize these differences so that treatment approaches can be tailored to specific groups of patients. As I mentioned, we also know that not all non-small cell lung cancers are alike. The same type of cancer behaves differently from one patient to the other. And we're beginning to understand why some patients do very well and some patients don't do very well with specific lung cancer subtypes. The biggest excitement, of course, is development of targeted therapies. There are many of them already in the clinical space now, and many, many more that are in the research setting, some of which are poised to enter the clinic in the next 1 or 2 years. We also have become better at combining chemotherapy with radiation to improve cure rate for patients with lung cancer that cannot be removed by surgery. These are what we refer to as stage 3 cancer patients. And if somebody has stage 3, the big question is, can you remove it by operation or not? If you can remove it by operation, we give them surgery, and then we give them chemotherapy afterwards. If, for a variety of reasons, the tumor cannot be removed by surgery, then we do chemotherapy with radiation, and that can cure those patients even though they cannot have surgery. Another big step forward is that we are using patients' tumor tissues to understand the genomic profile. What is the genetic makeup of the patient sitting in front of me or a physician? How can we use that information to treat that particular patient? I think this is now a mainstream approach for adenocarcinoma, and we're now beginning to use the same approach to squamous cell cancer. And finally, the most important good news among all is the fact that we can actually detect lung cancer early with CT screening. And by early detection, we can save lives, we can cure patients. Studies have clearly showed, or at least one large trial has clearly showed, that getting CT scans in patients with high risk, which is defined as somebody with history of smoking, age over 55, and does not have a prior history of lung cancer, by using CT scans, we can detect lung cancers. And this early detection technique will result in 20% reduction in deaths related to lung cancer. So now we have to implement CT screening for patients at risk for lung cancer, and we're quite early in the process. And hopefully Medicare and other insurance companies will start reimbursing patients for undergoing the CT screening. And I think that will really shift the percentage of patients diagnosed with lung cancer from being more advanced stage disease to detecting cancers in the earlier stage. That's what the success in breast cancer has been with mammograms: detecting cancers early, surgery, appropriate chemotherapy and radiation, higher cure rates. We need to do the same thing for lung cancer, and the data with CT screening is a very important step in that direction. Now let's focus about some of the advances in our understanding of genetic makeup of cancers. This is a cartoon that reflects what's called the EGFR pathway. This is one specific signaling network in the cancer cell. So in other words, when you look at a cancer cell, how is it different from a normal cell? The cancer cells proliferate, they can metastasize, which means they can go to other parts of the body. They don't respond to some of the checks and balances that are in our body. So what makes the cancer cell different from the normal cell? One of the main differences is some cancer cells have the EGFR pathway. This is think of this as an electrical circuit for the cancer cell. This electric circuit is what is keeping that cell alive. If you shut this signal off, that cell cannot do all the bad things that it does. So once we recognize that the EGFR pathway is an important signaling network for cancer cells, people started developing treatment approaches that specifically shut down this EGFR pathway. We refer to these drugs as EGFR inhibitor. These drugs were initially developed about 10, 12 years ago, and this is a very old chest X-ray from a patient treated with an EGFR inhibitor in the year 2002. And you can see here on the left is a CT scan, the chest X-ray showing a lot of cancer within the lungs. All these white areas are evidence of cancer. And this patient was started on an EGFR inhibitor, and just 5 days later you can see the chest X-ray showing a lot of improvement in the lungs. The whiteness or the cancer areas have significantly gone away, and this patient was feeling much better. And this story started emerging from patient to patient. However, this was only seen in about 10-15% of the patients who got these EGFR inhibitors. At that time, we did not understand what is it that separates those 15 to 20% of the patients who do really well with these drugs from the others who don't do very well. So a number of research groups started looking into that question, and now we have the answer. If you look at what is different for these patients, the answer is they have an EGFR mutation. So these are two papers that were published in 2004 that showed that these particular patients who have the mutation, this is a mutation that their tumor acquires. This is not something people are born with. The mutation happens sometime during the lifetime, and this mutation is the electrical circuit that keeps that patient's cancer alive. So if you shut that light off, that cancer will regress, and that's what we've seen with FDA-approved agents that are available in the U.S. and other parts of the world. And when we give these patients EGFR inhibitor, they do really well. Majority of them do. Their cancer responds considerably, their symptoms get better, and they go on to do well. And we have now learned that for a patient whose tumor has an EGFR mutation, the EGFR inhibitor is better than chemotherapy. So we don't go with chemotherapy for those patients, even when they have a lot of symptoms and they are not faring very well. So we give them an EGFR inhibitor. Now, what we learned is after a certain period of time, these tumors start growing back again. That's because they develop resistance. Now, what happens in a resistant setting is the tumor undergoes what we call a secondary mutation. So just like bacteria develop resistance to antibiotics, tumors undergo certain changes. So in a patient who has this mutation, which we refer to as the T790 mutation, the tumor basically changes the structure of the EGF receptor. So the drug that continued to work very well until that time can no longer sit and bind on this receptor site because the receptor structure has changed. So that blockade is released, so the tumor cells have the wires turned on again. Now, how do we address this situation? The good news in the past just 2 years, or even more recently in the past few months, is our knowledge that there are now new drugs that can actually turn off the resistant mutation. One drug is AZD9291. This is not FDA approved. This is still in clinical development. But you see here what we refer to as a waterfall plot. What it is every vertical bar represents the tumor size of a single patient. So here you see about 120 patients, and if the line goes below 0, that basically means the tumor got smaller, which means the tumor shrunk. If the line goes above, that particular patient's tumor got bigger. Now what you see here from this overall curve is vast majority of the patients, the tumor got better. These were patients who had received the EGFR inhibitor for mutation-positive cancer, did well for a while, then the treatment stopped working, and they were tested again with a biopsy, were found to have this new mutation, and got the newer drug, AZD9291, and are now responding very well. The response is nearly 65%, which means 65% of the patients experienced significant reduction in size of the tumor, and 95% of the patients benefited from these drugs. There is another drug which is called CO1686, which is also very similar to this drug, that is also being tested by clinical trials. And both of these drugs have now received the breakthrough status from the FDA, which means they are on a faster path towards getting FDA approval. So now we have ability to treat an EGFR mutated tumor with a specific drug, not chemotherapy, and after some time when resistance develops, we can do another biopsy, find out what caused the resistance, and if the patient's tumor has the T790 mutation that's seen in about 60% of those patients, we can give another specific drug which is tolerated remarkably well, very minimal side effects, and we see responses. So this is a big step in the EGFR world. Now what about if a patient does not have an EGFR mutation? We now know that there is about 5 to 8% of patients with lung cancer that have another abnormality called ALK fusion. And that's what is seen here. Basically, what happens here is there is a gene called ALK, which is normally silent. But for some patients, there is a rearrangement of this ALK gene, which wakes up this sleeping gene. And that causes the switch to turn on for the electric circuit. So just like EGFR for patients with EGFR mutation, ALK is for patients with ALK fusion gene. And when that is seen, specific ALK inhibitors are now available to treat these patients. Crizotinib is a drug that was approved by the FDA almost 4 years ago. And here again, you see that waterfall plot that we discussed a few minutes ago. When patients receive crizotinib, you can see significant reduction in tumor for a majority of the patients, and nearly 90% of them derive benefit. So this study formed the basis for approval of crizotinib. We now know even more than what we did 3 years ago about crizotinib. This drug is better than chemotherapy for patients who have ALK-positive disease. We also know that certain chemotherapy drugs work well for patients with ALK-positive disease, such as pemetrexed. So when we run out of targeted therapy options for these patients, we can treat them with pemetrexed, and that has good results. Just like we talked about for the EGFR mutation situation, patients' tumors, no matter how well they respond to crizotinib, unfortunately, at some point develop resistance. And we are now learning about specific mechanisms of resistance, and that is already leading to newer treatment approaches. Just about 3 months ago, the FDA approved a new drug for patients with ALK-positive disease. This drug is called ceritinib. It was also referred to as LDK378. This was given to patients with ALK-positive disease. As you can see from this waterfall plot, the blue lines represent patients who had already received crizotinib and unfortunately developed disease resistance after some time. And the yellow bars represent patients who had not received crizotinib. They were just newly diagnosed with ALK-positive lung cancer. And here again, you see the waterfall plot showing nearly 80 to 90% of the patients achieving some form of benefit with ceritinib. So this drug is now FDA approved, and the good news is there are at least 6 other compounds, very potent ALK inhibitors, that are being developed for patients with lung cancer. And what we are now beginning to see is very similar to what happened in the EGFR situation. You find out if the patient has ALK-positive cancer. If they have it, you treat them with crizotinib. At some point, their tumor will start growing again. Then you give them a drug like ceritinib, and you can get a response again. And those responses are also durable, which means they are not short-lived. They can go on for even a year or longer. Now, obviously, this is not cure at this point. We still have a long ways to go. But what we are seeing is the emergence of a paradigm. I just want to take you back to a different disease, which is called chronic myeloid leukemia. 10 years ago, or actually 15 years ago, that disease was treated with bone marrow transplantation. Then they found out it had a mutation. Then they developed an inhibitor that worked really well. Then after a while, it caused resistance. Then they had a second drug which will overcome resistance, and so on and so on. And now, by using these specific tyrosine kinase inhibitors, patients with CML rarely undergo bone marrow transplantation. They live for years and years and potentially can get these treatments on a chronic basis for 10, 15 years or longer. So we're beginning to see the first steps towards that development in lung cancer with these drugs. Now, another interesting development was identification of a new gene rearrangement. This is called ROS1. Just this week in the New England Journal, there's a paper about patients with lung cancer who develop ROS1 gene rearrangement. Just like ALK fusion, this is another gene rearrangement. A silent gene becomes turned on because of this rearrangement. And for patients who have this rearrangement, we know now that using even crizotinib, which is also a ROS inhibitor, results in significant responses in majority of the patients. So this drug, crizotinib, can be used to treat patients with ROS1 positive disease, and they respond really well. There are also other ROS inhibitors that are now being studied. Now, once we got to understand that there are specific mutations, people started looking for more and more. And what you see here is another mutation. Now, this mutation is only seen in about 1 to 2% of patients with lung cancer. We call this a RET fusion. The good news is if somebody's tumor has a RET fusion, even though there are no RET inhibitors approved in lung cancer, some of them are available for treatment of certain other cancers. So these drugs are already in the market. And we have started seeing successful outcome when these patients get RET inhibitors. So the idea now here is, as we discover more and more mutations, we're trying to match the mutation with specific drugs that the patients can get that maximize their chances of benefiting from these drugs. I want to point out that this discovery effort is only possible because of collaboration across academic institutions. The Lung Cancer Mutation Consortium is a 16-member academic consortium. You can see the institutions that are part of it, starting from the UCLA in the West Coast, Emory in Atlanta, Yale and Dana-Farber in Boston. All across the country, lung cancer investigators got together and started learning about specific mutations. So we collect the tumor specimens from patients, we test them, we conduct mutation testing, and find out what's the mutation that's driving a patient's cancer, and then work together to conduct clinical trials that can be subsequently form the basis for approval of new drugs. I also want to point out the critical role played by Free to Breathe. This is the administrative organization for the whole Lung Cancer Mutation Consortium, and they play a pivotal role in improving and promoting our efforts to understand specific molecular abnormalities in patients with cancer. Now, what did the Mutation Consortium do? Here are the results. If you looked at lung cancer about 10 years ago, it was one large pie. Everybody had lung cancer. We didn't understand who is who. Now we are beginning to piece this pie into different subsets. EGFR is 17% of patients with adenocarcinoma. You see ALK, and you see smaller and smaller pieces of the pie which are different mutations. But the bottom line is nearly 2/3 of the patients, 64%, in the Lung Cancer Mutation Consortium effort were found to have what we call now a driver mutation, a specific gene that is driving that patient's tumor. Now, if the patient's tumor has a mutation for which we have a readily approved medication, we give that medication to them. If there is no FDA-approved medication, we try to match them to a specific clinical trial that maximizes their chances of success because of these efforts. So when we have a patient with lung cancer right now, our approach is broken down this way. We see whether they have a known driver, and if they have a driver that's targetable like the EGFR, ALK, or ROS with existing treatments, we give them targeted therapy. If there is no driver mutation in that tumor, then we treat those patients with chemotherapy. And if there is a known driver but there is no proven therapy, we try to match them to specific clinical trials. So let's now look at what's happening with squamous cell cancer, which is the other subset of lung cancer that's common. nearly 30 to 35% of patients with non-small cell lung cancer will have squamous cell cancer. Just like we did with lung cancer, there are now research efforts that are looking at specific mutations in patients with squamous cell. And we are already coming up with specific mutations. This particular mutation called PIK3CA mutation is seen in about 16% of these patients. Another abnormality called FGFR amplification, that's seen in about 10%. So we're beginning to break the squamous cell patient group into specific subsets and therefore develop treatment options for them as well. And national effort for these patients is, sorry, I skipped a slide. I'm going to go back. Is what we call the Lung Master Protocol. This clinical trial is now available. This is a collaborative effort including many groups, the cancer cooperative groups, the NCI, private foundation. Free to Breathe is part of this effort supporting it. Basically what we're doing is for patients with squamous cell lung cancer, we do mutation profiling of their tumor. And if they have a specific mutation, then they are enrolled to a clinical trial where they'll get a specific inhibitor. And you can see here that there are already 4 different mutations for which there are clinical trials. And we're now working on adding more and more arms to this trial that will cater to potentially every single patient with the squamous histology. Another exciting development we've seen is in immunotherapy landscape. Here you can see that immunology is important for a tumor. This is what we call a nevus. So this is a mole, and if you can see what happens here is there's a circular rim of whiteness of this mole. And when one starts seeing the circular rim, it's only a matter of time before the mole is eaten up by the immune system. So this rim is nearly something that heralds that the patient's immune system is attacking that mole, and it's going to be able to gobble it up. So why can we not use immune therapies to treat tumors? This has been a pursuit for a long time, but we're beginning to see success. This is a drug called PD-1. What this PD-1 does is makes the patient's immune system not see the tumor. So they are active. These are readily charged to fight the tumor, but they cannot see it. Now by using antibodies, we can actually make the tumors visible to these immune cells, so these patients can subsequently benefit from activation of their immune system. Here is a patient I treated in my clinic with a drug called nivolumab. The patient had 10 to 15 masses just like this, tumors in the lung. This was July 2013, and 6 months later, you can see that the patient's tumor has gone down considerably, and there was a dramatic response in many of these tumor spots, which shows us that the power of immune therapy is here to help us. So a number of drugs that target the human immune system are now being developed in lung cancer, and we anticipate that in the next year or 2, these will be available even in the routine clinical setting. So those are some of the advances, but we clearly have challenges to face. Resistance, how do we manage resistance to targeted agents? Maybe combination approaches, like how we treat HIV now. No one drug can cure HIV, but a combination of drugs can result in long-term survival with HIV disease. That's what we need to develop for lung cancer for targeted therapies, and we're already beginning to do that. Metastases, emergence of metastases is a major problem. We need to address that because we can handle what we see in the patient's scans by using surgery or radiation. It's often what you don't see that results in recurrence of cancer. For example, if you look at stage 1 lung cancer, only 75% of the patients are cured. Why can we not cure 100% of them? That's because metastases happens early. And these patients who recur, they occur not in the lung, often in the brain or other sites, because these tumor cells have already escaped and moved to these organs. And now we know that chemotherapy after surgery results in improved cure rate, but we still are not where we want to be, which is 100% cure rate. We're now conducting trials that try to address improving chemotherapy. Here is an approach of a clinical trial where an anti-angiogenic drug, a drug that cuts off blood supply to the tumors, which is critical for them to survive, is combined with chemotherapy. We have already treated nearly 1,500 patients on this study, and we hope to have the results in the next year or so to see if adding an anti-angiogenic drug results in improved cure for patients with early stage lung cancer. Another approach is what we refer to as the alchemist. This has just started. What happens if a patient's tumor has an EGFR mutation? You've already removed the tumor by surgical resection. Now there is a randomized trial that tries to give them an EGFR inhibitor after surgery to see if that can actually cure those EGFR positive patients. We also have a trial for ALK positive patients. After surgery, you give them crizotinib to see if that can be cured. So we're now beginning to take these targeted agents from treatment in the advanced stage disease to earlier and earlier in the course of treating lung cancer. These trials will require a collaborative effort among academic institutions, patient community, and other supporters in order to get them done so we can find out how these help cure patients. And therefore, it's important for us to increase the awareness about the availability of these trials. Now, last part of my talk, in the next couple of minutes, I'll finish up saying what are the major challenges with lung cancer. Metastases. By the time we find out somebody has cancer, as we talked about, a lot of microscopic cells have already escaped out of the primary tumor and are hiding either in the brain, the bones, the adrenal glands which sit on top of our kidneys, the liver, and the lymph nodes. So we have to understand better how tumors metastasize. It is strongly influenced by the tumor and its neighborhood, what we call the stroma. We haven't paid a lot of attention to the interaction between the tumor and the stroma in the years of research. We need to understand that better. We know that the stickiness of a cell and the mobility of a cell are critical for cancer. How can we turn it off? How can we make the cancer cell not metastasize? What genes are involved in turning the tumors to metastasize? These have to be understood better. One example I can show here is a gene called LKB1. This is seen in nearly 25% of the patients with lung adenocarcinoma. There is a mutation in this gene, and patients whose tumors have these mutations end up with more metastases. Why do these patients have more metastases? How can we block this metastasis from emerging if you know a patient has an LKB1? So these are things that we need to pay attention to, and I think that will result in even better therapeutic gains over what we've seen. I'm very pleased to hear and have been part of the Free to Breathe initiative to promote research to focus on preventing lung cancer metastases. And as Annie will tell us, the organization has taken up the goal of doubling the lung cancer survival rates by the year 2022. And in order to achieve that, promoting understanding about metastases and developing ways to prevent metastases are critical. So what have I said so far in this presentation? There are significant improvements that have been made in the treatment of lung cancer. Survivals have improved for every stage of non-small cell lung cancer. Cure rates have improved for early stage disease, but we still have a long ways to go, even for patients with those specific mutations where cure is still elusive. We need to continue our research efforts. The previous successes did not come by chance. They came about because all of you and other organizations increased the awareness about lung cancer, increased funding to lung cancer. We're nowhere near where funding should be, but whatever funding increments we're seeing have resulted in better research and better treatment outcomes. And we need to keep our foot on the gas pedal so that the discovery and accelerated treatment discovery efforts go on unimpeded so we can come to an age where lung cancer is a treatable and curable disease. Thank you so much for your attention, and I'll be happy to take questions. Let me turn it over to Annie at this point.
Thank you, Dr. Ramalingam. Before we move into our Q&A portion of today's webinar, I wanted to quickly share, as Dr. Ramalingam referenced, Free to Breathe is working to double lung cancer survival by 2022, and we can't do that without the work of people out in their communities helping to fundraise and support our research grant programs. An opportunity that all of you can consider by asking your communities is to help us by donating today or joining an event or fundraising for us. You can go ahead and donate today at freetobreathe.org support.org/donate and help us work towards our vision of doubling lung cancer survival. Help us to fund really exciting research like Dr. Ramalingam referenced today. And once again, I want to thank our generous supporters, Lily and Beringer Ingelheim. Before we move into Q&A, I did want to reference that we will be providing follow-up recording of this webinar, so you can look forward to seeing that email within the next couple days. We've been receiving a number of questions through our chat feature. Please feel free to continue bringing those questions to us. We're going to address as many of them as possible. Dr. Ramalingam, we have the first question for you. We're starting to see a lot more young people who've never smoked getting diagnosed, and even at end-stage lung cancer. Can you help us understand why that might be?
So that is a very important question. Nearly 10 to 15% of patients with lung cancer we see now are less than the age of 40 years, which flies against the notion that lung cancer is seen after somebody has been exposed to risk factors for a long time in their lifetime. We understand biologically that some of these patients are more likely to have certain mutations, and these mutations are probably what are contributing to the lung cancer. We still do not know what causes this mutation. What we know is patients are not born with these mutations. A lot of my patients ask me, since I have this mutation, does this mean there is a higher risk for my kids or my siblings? And the answer is no. These mutations are not something one is born with. They develop during the course of somebody's life. And we're just beginning to understand lung cancer in never smokers. A lot more work remains to be done.
Thank you. Another question involving women and lung cancer. Do you think that there's any specific reason that more and more females are receiving a diagnosis, and is there a difference in treating men versus women?
This is something that we're seeing more and more. Unfortunately, women being diagnosed with lung cancer is becoming more common. The good news is the biology of lung cancer women is now beginning to be understood. We're trying to see what role the estrogen hormonal milieu plays in lung cancer. There are differences in how patients with lung cancer behave based on their sex, men versus women. Certain treatments appear to work a little better in women than in men. Survival in women tends to be better compared to survival in men, stage to stage. So there are differences. We are beginning to understand these differences. We also know that women are at higher risk for developing cancer following tobacco exposure compared to men. If you had a man with a comparable tobacco exposure to a woman, the woman has a higher risk of developing. That goes to some inherent ability of the body to repair DNA damage that occurs after smoking cigarettes. So a number of differences there. We have explanation for some, but not all of them.
Thank you. Another question, can any of the drugs that you referenced when you discuss targeted therapies, can they be used once the cancer has metastasized?
Right now, actually, the targeted therapies are primarily used after metastases is developed in advanced stage of the disease, or stage 4, as it is referred to. Our goal is knowing how well these drugs can work in the metastatic disease or advanced stage setting. By moving them to patients with early stage disease, we can actually use these tools even more effectively than we are using and use it to improve the cure rate rather than trying to play catch up in the advanced stage disease. So those are the trials, some of which I alluded to earlier on.
Do you know if there are any targeted therapies out there for small cell lung cancer?
Small cell lung cancer, unfortunately, we have not made much progress in the past 20 years. Part of that is because we did not understand very well the genetic makeup of small cell lung cancer. In the past 2 years, people are beginning to look at genomic characteristics of small cell lung cancer and try to use targeted agents. These are very much experimental at this time, quite early I would also say, but knowing that this is the path that has led us to relatively successful outcomes in non-small cell lung cancer, we're going to see more and more of these approaches in the very near future.
Thank you. Another question. A number of patients with the EGFR mutation don't respond to gefetinib or erlotinib, suggesting that additional features may be involved, like KRAS or NRAS. What is known today about additional mutations besides EGFR?
Sure. That's a very good question. What we've seen is about a third of the patients don't respond well to gefetinib, erlotinib, or afatinib, even if they have an EGFR mutation. KRAS mutation is not seen in the same patient with an EGFR mutation. The cancer cells are so smart, they pick one pathway to activate themselves. They don't need two pathways. So if a tumor has an EGFR mutation, you don't see a KRAS mutation. But what we have now learned is that there are certain other genes that the cancer cells may take advantage of. One of them is a polymorphism in a gene called BIM, and that at least in Asia, they've reported that if patients have certain variations in that gene, they don't respond quite as well to erlotinib and gefetinib. So that subgroup of patients, we need to develop new treatments in addition, or combination strategies with the EGFR inhibitors to better control their cancer.
Thank you. Another question, if somebody is not ALK or EGFR positive, when is that testing done for mutations, and are they now testing for additional mutations such as ROS1? Is that done for every lung cancer patient? There was one person in particular who mentioned that by the time they found out that their daughter was ROS1 positive, it was too late for any treatment options.
I'm really sorry to hear about that particular situation. I think we need to do molecular testing at the time of diagnosis for patients with lung cancer, so our patients who have these abnormalities can take advantage of these treatment options. The standard of care in patients with lung adenocarcinoma, and I would even extend it to anyone who has non-squamous cancer, at the time of diagnosis of advanced stage disease, we need to do mutation testing. The testing technology is available. It's not expensive. It can be done in many hospitals throughout the country. There are commercial entities that offer this testing. So I would strongly encourage my colleagues, physician colleagues, to do genetic testing at the front end as soon as the diagnosis is made. I realize there are certain situations where you don't have the 1 to 2 weeks it takes to get the genetic testing results before you start treatment. So in those situations, what I tell them is, go ahead, start chemotherapy, but get the results right away. As soon as you get the results, based on how the patient is doing, use that information to change therapy as needed. But we need to be doing molecular testing for every patient with advanced stage lung cancer that has non-squamous lung cancer.
Another question about a specific participant's family situation. One of their family members has stage 4 EGFR mutation, and she had a brain scan, but the doctor didn't follow up. Would a scan show what you referenced as the hiding cancer cells?
The brain tends to be a place that we refer to sometimes as a sanctuary site because it is not an area where chemotherapy and other medicines can readily penetrate. MRI of the brain is good at picking up spots that are a few millimeters, but they cannot pick up microscopic cells. So if somebody has symptoms that would lead us to think that that person has a brain spot, we get an MRI scan. In somebody who does not have any symptoms or signs on exam, then we don't routinely get MRI of the brain. We rely on the treatments to do their best and follow the symptoms and signs closely.
Thank you. A couple questions about the EGFR mutation again. If someone was found to have EGFR or perhaps a different mutation a couple years ago, should they have their tumor retested for additional mutations?
That is where we're increasingly going towards. With time after treatment with targeted therapies, tumors develop new mechanisms of resistance. So if we were going to treat them in a more informed manner, we need to understand what changed between now and from 2 years ago. So testing these mutations, again, doing another biopsy, while it is not easy for our patients to go through biopsies, unfortunately, until we find out other ways to test for these mutations in peripheral blood, we need to resort to these biopsies.
Dr. Ramalingam, what causes mutations to occur in the first place?
Well, in patients who have history of smoking cigarettes, it's the carcinogens in the tobacco that cause mutations. Like, for example, the KRAS mutation, which is seen in nearly 25 to 30% of our lung adenocarcinoma patients, is caused by exposure to cigarette carcinogens. In EGFR mutation, we do not know what causes it. That is a big mystery until now. But each of these mutations, majority of them in smokers, are directly related to exposure to cigarette smoke. In never smokers, that's a question we have not answered yet.
Okay, thank you. Is there a drug in trials as effective as Tarceva but that has less toxicity?
What we refer to now as the third generation EGFR inhibitors, such as the CO1686 and the AZ9291 compounds, these are drugs in clinical trials. They're not FDA approved. They are now being developed for patients who develop resistance to erlotinib and gefitinib. These drugs are very specific for the mutated receptor, so their toxicity profile or safety profile appears to be much better than what we see with erlotinib or afatinib. But it's a little too soon to start using them before somebody has seen erlotinib or afatinib, because we do not understand what's the best sequence to give these drugs in.
What type of adjuvant therapies do you recommend after chemo and radiation have been given?
So in patients with stage 3 disease, which is not removable by surgery, we treat them with chemotherapy and radiation. And after the treatments are complete, we don't continue the treatments. We just enter the follow-up phase where we look at scans every few months or so. In patients whose tumor is removed by operation, we give them what is called adjuvant chemotherapy to try and eradicate those small microscopic cells that may be hiding. Radiation in the adjuvant setting is used in specific situations, not for all patients. But once you complete that adjuvant chemotherapy and, if needed, radiation, we just follow patients with scans. There's no need to keep them on treatment, at least as of now, forever.
Are there any treatments that combine both chemotherapy and immunotherapy drugs in trials at this time?
That's a great question. The promise that we've seen with immunotherapy is clearly leading in that direction. The trials that are ongoing now are trying to see how safe it is when you combine an immunotherapy drug with chemotherapy. And at least the initial returns have been promising. And then we now will move to the next phase of research where we will try to understand how effective the combinations are. Once we learn that, I think there is a great potential for chemotherapy and immunotherapy. Sometimes the best answer may not be the combination. It may be sequencing. You give chemo first and then follow up with immunotherapy, or you give immunotherapy first and follow it up with chemotherapy. So those trials are also ongoing to see how immunotherapy fits into the existing landscape.
Thank you. One person is asking, what's the percentage of EGFR patients who have to transition to T790?
At this point, we estimate that approximately 50 to 60% of the patients will end up with the T790 mutation. These are patients who start off with an EGFR mutation, they're treated with an inhibitor, and develop resistance. So that's the patient group that would stand to benefit potentially from the third generation T790 inhibitors that are in clinical trials that we talked about.
Dr. Ramalingam, what are you most excited about that's in the pipeline for lung cancer research?
Can you please repeat that question, Annie? I missed the last part of it.
Yeah, I'm wondering what you are most excited about that's in the pipeline for lung cancer research.
Sure. So there are a number of areas that excite me. One is for EGFR landscape, the third generation drugs are exciting because of how effective they are and how well tolerated they are. And we think that those can form the basis for developing new combination approaches. A lot of us see combinations as the way to prevent resistance or delay resistance. So that's an area of exciting development. The second thing is we're also now increasingly learning to use peripheral blood to check for mutation profile. The technology is a little bit early, but I think with the next 2-3 years, we'll be using peripheral blood to look for specific mutations in the routine care setting. That would make it life easier for patients because repeat biopsies would be less and less necessary. In the ALK patient population, there are at least half a dozen exciting drugs that are very potent that are in the clinical trial setting that we're all excited about. Immunotherapy is another area that I mentioned where these drugs are helpful in shrinking the tumor of patients, and not only that, some of these patients go on for 1-2 years without any evidence of disease progression. I have a patient who just got 1 dose of an immunotherapy drug and unfortunately could not continue, but it fortunately turns out that a year and 4 months later, that tumor has completely disappeared and this patient's tumor has not recurred. Now, we don't know why it is that this patient had a dramatic response versus others, some of whom who did not respond. That's where we need to focus our efforts on. So many exciting developments, many new drugs in clinical trials, but to name a few, the new generation targeted drugs, immunotherapy drugs are some of which I'm most excited about.
Great, thank you. Someone is wondering if it's possible to cure someone with an ALK mutation.
At this point, we are using the ALK inhibitors for patients with advanced stage disease. So cure has not been an outcome that we've seen. Patients' tumors develop resistance. But what we are doing at the Eastern Cooperative Oncology Group is a clinical trial in ALK-positive patients who have early-stage disease. So these are stage 1, 2, 3 patients. After surgery, we're giving them an ALK inhibitor to see if that can improve their likelihood of cure. So this trial will take a handful of years to accrue and complete. So we may not have an answer in the near future, but my hope is that such an effort will bear fruit for our patients.
Dr. Ramalingam, could you speak to if the screening CT scan, if it's different than a diagnostic scan?
Sure. So what we do for screening is referred to as a low-dose CT scan. This involves a lower exposure to radiation, and it's a single breath hold. In other words, the patient holds the breath for a few seconds, they acquire the images, it's done. The usual diagnostic CT that we do involves more frequent sections, cross-sections. So these are what we refer to as 2 or 3 millimeter cross-sections. So it takes a little longer to acquire the images. They also provide higher quality information, but for screening purposes, we don't need that level of detail. All we need to know is there something wrong or not? If we find out something is wrong, then we can resort to other study to see what is wrong and how do we address it. So that's the main difference.
Thank you. Another question. Folks with the EGFR mutation or the ALK mutation have more options for treatment now. But what about the people who have small cell lung cancer or squamous cell lung cancer? What would you say to those folks? Are there exciting treatments on the horizon for them?
I think the good news is, at least in squamous cell cancer, we now have entered the genomic era where we are sequencing these patients. At this time, it's still experimental, but the clinical trial that I talked about, which is a large national effort, is trying to do just that. Hopefully, we'll come up with some good hits like the EGFR and ALK in squamous patients. In small cell, unfortunately, we are not as far ahead as we should be. We're starting, though, and with the collaborative efforts and greater emphasis on funding small cell lung cancer research, my hope is that we will be able to make significant dents against this disease.
Great, thank you. And one final question. Could you speak a little bit to the importance of clinical trials and how that advances lung cancer research?
This is one of my favorite topics, which is how important clinical trials are to improve outcomes for patients with cancer. We can have all these exciting drugs in the laboratory, but none of them would be able to come to our clinical setting unless we test them in a carefully controlled clinical trial. There are a lot of myths about clinical trials, a lot of false information out there that we all need to correct to help our patients make decisions. But participation in clinical trials helps not only move the field forward, but potentially helping the patient get a treatment that their tumor needs. that may not be available in the routine setting for a few years. A good example is the T790 inhibitors. These drugs are not in the market at this time. They're only available through the clinical trial. But when you give it to a patient who has developed resistance to erlotinib, they respond dramatically. So these are the reasons why we need to keep thinking about clinical trials, encourage participation whenever possible. I realize there are many situations people may not be able to participate. Right now, only about 3 to 5% of patients diagnosed with cancer, adult patients, enter clinical trials. If we just double that from 5 to 10%, we can bring drugs to the clinic so fast. In the past, it used to take 10, 15 years before a drug entered the market or the clinical space. Now, as we saw in the ALK story, the research done 4 years ago results in a new treatment. So we're shrinking the time it takes to develop drugs and bring them into the clinic. So what is happening today will make an impact just not 15 years from now, but hopefully in 3 to 4 years. And participation in clinical trials and promoting clinical trials will help us get there faster.